Thank you. I want to thank the organizing committee for giving me the opportunity to put on this work, our work. This work focused on the duplication in phages and to the description of a new phage recombinase family. These enzymes have been classified into three main superfamilies in phage. The WET52-like protein with the well-known example of WET beta, which is encoded by the lambda phage and which is used in a genome engineering methodology called recombinating. The WECA-like superfamily with full-length or coronally WECA homologue. And finally, the Gp2.5-like family, composed to date of strict homologue of Gp2.5 from the T7 phage. These enzymes are dedicated to homologous recombination, which is a mechanism allowing the repair of double-strand break on the DNA, following two main mechanisms, the single-strand invasion and the single-strand annealing. Most of the recombinase phage to date have been involved in this kind of pathway, single-strand annealing, but WECA full-length homologue are able also to strand invasion. So depending on the mechanisms, depending on the recombinase used, and depending on the template, the repair can be faithful, denoted by homologous recombination, but also can lead to chromosomal rearrangements such as duplication, gene acquisition, and deletion. And this is one of the bases of the phage evolution. These enzymes are also required for optimal formation of virions. Some of them are strictly required for this. And more recently, some of them have been involved in resistance to anti-phage system. So clearly, recombinase in phage are important proteins for the life cycle of these entities. And then it is striking that in our previous study in 2010, only approximately 50% of phage genome encode a known recombinase. And we have two main explanations. Some phage are able to use the recombination machinery of their host, but we propose that it remains to identify uncharacterized recombinase. So to search for recombinase, one way is to search for recombination events. And we built a study, started from a study from Laurent de Barbier's group. We have shown that pitane, which is a phage infecting LF82 Escherichia coli strains belonging to the Felixounavirus genus, is able to change its host range in the gut of conventional mice, and this is due to a point mutation in one of the two-tail fiber genes encoded by these phages. Interestingly, among the phages that have evolved and changed their host range, an event of duplication could be shown here with the duplication of the red cassette between these two tail fiber genes. The spacing between the duplication and the fact that there are borders here with a sequence in green by short imperfect repeats strongly suggested that the recombination event has been mediated by a phage recombinate and not by the host RECA recombinase. However, there is no trace of recombinase in the genome of pitane. So before to make an in-depth characterization of the genome of pitane, we wondered if these duplication are common in phage. Then we wonder if there is a relationship between the presence of duplication and the presence of recombinase in phages. And then we search for recombinase in this phage. So to search for duplication, we developed bioinformatic tools named FRUB for finding repeats using BLAST, using the following parameter: duplication that are higher than 100 base pairs to avoid tandem repeats that can be arrived by polymerase slippage, with an identity of more than 85%, and not present at the phage ends to eliminate tandem repeats that can occur for some phages. We used a dataset of more than 5,000 complete phage genomes available in an infrared database, and we have annotated this genome using Pharoka. And we have found that approximately 23% of phage genomes contain duplication. When we localize this duplication on the genome of the phage, we find that they are enriched in the gene coding for tail protein, here in blue. Indeed, 1.8% of tail genes contain at least one or more than one duplication. And this is the most duplication enriched gene category. So duplication in phage genomes are frequent and enriched in tail genes. We have updated our knowledge about the content in recombinase in the genome of phage. So in our data set, using the FROG database. So FROG is a database of more than 800,000 phage protein classified accordingly to their sequence homology and their structural fold. We have found that more than 60% of phage encode recombinase, which is 10% higher than our previous study. We have found that they are most prevalent in virulent, 62% relative to the temperate with 58%. And we also seen that the family of recombinase is different between the two lifestyles, with an enrichment in WET52-like protein in blue here in temperate, and an enrichment in Gp2.5 and UVSX in the virulent compared to temperate. So the content is different between the lifestyle, and because we observe these differences, we included this parameter in our following analysis, aiming at the comparison of between recombinase and duplication contents in phages, in virulent and in temperate phage. Here you can see the percentage of phages relative to the number of duplication in genomes. And we observe that virulent phage with recombinase here in red are always overrepresented compared to the phage without recombinase in blue. And this is the opposite for temperate phages. So we have found that 53% of virulent coding for recombinase contain a duplication, which is significantly higher than the 43% without recombinase, and the opposite for temperate. So we conclude that the presence of duplication and recombinase is correlated in virulent, but seems anti-correlated in temperates. To gain a broader view of these events, presence of recombinase and presence of duplication at the genus level, we classified our 5,000 genomes in 95 genera, accordingly to the classification of the infrared database. These genera contain at least 10 genomes to avoid special case, and we check for the presence of duplication and recombinase in these genera. So here you have the 10 most enriched genus in terms of duplication, with the example of Felixounavirus and pseudo-T-even virus that contain phage that have all at least one duplication. And we have indicated also the content in recombinase with the color code depicted here. And strikingly, we found that only in Felixounavirus, phage do not encode for non-recombinase. This strongly suggested that this genus, phage in this genus, may encode an unknown recombinase. So to search for this recombinase, we use tools allowing identification of remote sequence homology, such as Phagonote or HHPRED. We screen for all the 66 predicted proteins from P10, which is a Felixounavirus. And we found only one, Gp64, sharing similarities with recombinase. These proteins belong to the FROG 3877, composed of protein from Felixounavirus of unknown function, but clearly linked to the FROG 112, composed of Gp2.5-like proteins. And we also see that there is a hit for the Gp2.5 from T7. We confirmed this remote homology at the sequence level by analyzing the structure of the protein. So we predicted the structure of the protein using alpha fold Swiss. The structure is presented here with a good accuracy, at least for the N-terminal part here in blue, showing a beta barrel fold. We asked for the presence of structural homologue in the database using fold-seq based on the predicted structure, and we found two Gp2.5 protein, one from the ANC34 and one from the T7 phage, and their structure are presented here with a characteristic oligonucleotide oligosaccharide binding domain here, which is involved in the single-strand binding of the DNA, and in its annealing ability. So when we overlay these two proteins, Gp64 and Gp2.5 for BT7, we observed a good overlay at the level of the OB domain of Gp2.5 with an RMSD of approximately 1 angstrom, but the rest of the protein differs structurally with an RMSD higher than 20 angstrom, raising the question of the ability of this protein to promote recombination event. So to test this, we used an recombination in vivo assay, which is called ssDNA recombination, in which we use the histological restraint that is chloramphenicol sensitive. And we electroporated these cells using an oligonucleotide, which is if it is able to integrate at the level of the replication fork thanks to a recombinase, correct two-stop mutation in the gene coding for the acetyltransferase and render the cells chloramphenicol resistant. We have shown that expression of Gp64 alone allows a significant increase of about 7-fold compared to the control of the recombination frequency, control without phage recombinase. So we conclude that Gp64 is a new recombinase. However, with a weaker activity compared to the red beta protein from the recombination module of lambda. Interestingly, Gp64 is encoded by a gene which is upstream of a gene coding for an exonuclease, and we wonder if these two proteins can promote recombination event using double-strand and not single-strand as in our previous assay. So we use the PCR product here instead of the single-strand here. And we observe that expression of Gp64 or Gp63 alone does not allow a significant increase in the recombination frequency compared to the control. However, the co-expression of the two genes leads to a significant 20-fold increase of the recombination. So we conclude that Gp64 works with its exonuclease partners to promote recombination event. However, here also with a weaker activity compared to the module of recombination from lambda. And then confirming the relationship between the presence of a recombinase and duplication that have been shown in this phage. So to conclude, we have found that duplication are frequent in phage and more common in tail genes, and we propose a role in host range evolution, as that has been known for the P10 phage in the previous. We have shown that more than 60% of phages encode an identified recombinase, but we propose that other recombinases remain to be discovered because other phage genus are enriched duplication, and we have no known recombinase in their genome at this moment. We have found a positive correlation between recombinase and duplication in virulent phage, but not in negative, but negative in temperate. So we have three main hypotheses. This difference could be depend on the nature of the recombinase. We have shown that we have a content in terms of family, which is different between the two lifestyle. Duplication are a known source of rearrangement in the bacterial chromosome. So having a temperate phage with duplication in the chromosome could lead to a deleterious effect for the survival of the bacteria. And we also propose that the host change may be more important for the persistence of virulent phage in their environment, because simply if temperate can be stably integrated in the chromosome of their host and then can propagate in their environment, which is not the case for virulent phage. And finally, we characterize a new family of recombinase in Felixounavirus, which we called GlaF for Gp2.5-like annealase in Felixounavirus, that works with its exonuclease, forming a new like exopair, as observed in the RED52-like family, superfamily of recombinase. So to finish, I would like to thank all the people that have been implied in this project. So Marie-Agnès Petit and Olivier San in our team, Laurent Debarbier and Alexis Cosiolo, Louisa de Sordi from Sorbonne University, and finally Kantalem Besni, which have done a large part of this work concerning the bioinformatics. And thank you.